I've been in Adimab for about a year. So in Adimab years, that's pretty short. So I'm going to give you a really nice overview of all the really cool things I've learned in my first year at the company. Okay, so if you're not familiar with Adimab, we are a premier therapeutic antibody discovery company, so we're really focused on discovering therapeutics for our partners. We're based in Lebanon, New Hampshire, where our primary labs are based. We also have a small site in Mountain View, California as well, where our computational team is based. That's also where I'm based as well. We're still a privately owned company. We were founded in 2007, and then we started our first therapeutic partnerships in 2009. Since that time, we've worked with over 130 different partners, and as I said, we're really focused on really working with our partners to discover therapeutic antibodies, and with those 130 partners, we've really haven't done any work on our own internal pipeline, and we've helped them advance over 80 molecules into the clinic with those 130 partners. And with that, 5 of them have advanced onto the market, soon to be 6. So one of my favorite books as a child was Charlie and the Chocolate Factory. And when I joined Adimab, I was really reminded of this book because if you're not familiar with the book, Charlie had this amazing opportunity to get a tour of Willy Wonka's factory. And so when he got this amazing opportunity, he got to see all of these amazing rooms in the factory, and shown here on the right is a map of the factory. And the reason why I was reminded of this book when I joined Adimab is that similar to Charlie, when he got to see all these amazing rooms and all these amazing inventions of Willy Wonka, I was also kind of blown away by all the amazing technologies and all these cool inventions that I got to see in my first year at the company. So I actually wanted to use this as a framework for my talk today, and I created my own map of what I'll call the Adimab factory, where I actually made 10 different rooms which correspond to 10 different technologies that I really want to highlight in my talk today. And I also created a little chevron across the bottom which will highlight those 10 different rooms as I kind of go through it. I'm actually going to spend a lot of time in the very first room, which is our full IgG HCAB presentation workflows. So I'll probably spend about 10 minutes in that very first room. Okay, so let's get going. All right, so for that very first room, these are our synthetic libraries that are in our proprietary yeast. Again, we're a yeast presentation company, and so I'll talk a lot about our yeast in a second. And we have both IgG as well as HCAB or VHH libraries that we present on these yeast. And for our IgG libraries, we have both kappa as well as lambda libraries, and we also have some common light chain libraries as well. These antibody libraries, these IgG libraries are heavily germline based with most of the diversity in heavy chain CDR3 with a little bit of diversity in light chain CDR3 as well. And on the next slide, I'll show kind of the length diversity in those libraries as well. The overall diversity of all the libraries, both the IgG as well as the HCAB libraries, are on the order of 10 to the 10th. And if you're familiar with yeast-based libraries, 10 to the 10th, that's pretty diverse. It's oftentimes really difficult to make a 10 to the 10th library in yeast, and that's really due to the fact that we can create really highly diverse libraries in our proprietary yeast. And then in terms of our common light chain libraries, we do have a common light chain library where we utilize our light chain for our CD3 IgG binder, which I'll talk about later in a subsequent room. And then we also have a common light chain library where we utilize 9 different germline common light chains as well. And then I'm not going to talk a lot about our HCAB library because I'll actually spend a dedicated room on our HCAB library on a subsequent slide. Okay, and then as I promised, I also wanted to talk about kind of the length diversities of these IgG and HCAB libraries. For the IgG, kappa, and lambda libraries, the heavy chain CDR3 length is on the order of 16. We also have some boutique kappa libraries where we also have some shorter as well as longer heavy chain CDR3 lengths that we can utilize for some different types of targets. We heard in the last talk around GPCR targets that maybe sometimes you might want to use a longer heavy chain CDR3 length we might want to utilize for targets like that. And then for our HCAB library, as I'll show later, those libraries actually have a slightly longer heavy chain CDR3 length. Okay, so in terms of, again, diving more into the overall design, as I mentioned, I actually sit in Mountain View with our computational biology team. We spent a lot of time really designing our synthetic libraries with that team. We fold a lot of immunogenicity as well as developability considerations into the design of that library, primarily in that heavy chain CDR3 as well as light chain CDR3 region of the library. What I also didn't mention, again, on that previous slide is that we use a lot of heavy chain germlines as well as light chain germlines in the construction of those libraries as well. As I mentioned, the overall diversity of all those libraries is on the order of 10 to the 10th. But when we synthesize both the IgG as well as HCAB libraries, we synthesize them on the order of 10 to the 12th. The reason why that's important is that we actually make multiple copies or diversity sets within that 10 to the 12th diversity. So that allows us to create multiple copies of each of these libraries, and each copy has its own set of diversity. So the reason why that's important is that if we have multiple partners that come to us with the same target, we can pull out a different diversity set so that we never have to select the same library twice against the same target, so that we never have to deliver the same antibody twice against the same target. So that's really important moving forward, and we never really have to ever go through gatekeeping as a result of that. And then, as I mentioned, as we'll talk later, we really pride ourselves on developability, and we fold that into our overall library design. And then, as we'll really stress later, our proprietary yeast is really the centerpiece of our overall technology. It allows us to present the full modality on the surface of our yeast, so the full IgG or the full HCAB. And then also, a really nice attribute of our yeast is that we can easily switch from presentation to secretion as part of our overall workflow. Okay, so let's get into the overall workflow and how we discover antibodies from our overall libraries. So we can take our biotinylated antigen, we can mix it with our libraries that I just showed in the previous slides, and then we can use a combination of max and fax-based selections to sort out those populations on the sorter. And again, we can sort for both expression as well as binding. In the later rounds of selection, we can also do deselections for poor developability. Adimab is actually very famous for utilizing what's called a polyspecificity reagent, which is a CHO membrane prep, where we can actually deselect for antibodies that have polyreactivity or stickiness, and so we can actually deselect for those antibodies from our selection. And so after we do multiple rounds of selection and select out those antibodies that have the right profile, we can then pick a small set of clones to then express and purify. As I mentioned in the previous slide, we can easily switch our yeast from presentation to secretion, and we can then express and purify typically up to 96 antibodies to then make and test. And that characterization results in a really rigorous data package that we present back to our partners, which typically includes kinetics, epitope binning, cell binding, and a whole host of other really rich data package back to our partners. So one thing I didn't mention in the previous slide is typically, one thing that we typically do in the later rounds of selection is what we call a light chain batch shuffle for our IgG selections. And as I mentioned, most of the diversity in our library is in heavy chain CDR3, so by doing a light chain batch shuffle, we allow the heavy chains to see additional light chains. And for our HCAB libraries, we typically do what's called a heavy chain CDR1-2 batch shuffle, allowing the heavy chain CDR1s and the heavy chain CDR3s to see additional heavy chain CDR1s and 2s in the HCAB selection. So that allows for additional diversification in the overall workflow. In that primary discovery workflow with our partners, it takes about 4 months. And shown here on the right are some typical affinities we see for both the IgG as well as HCAB workflows that we typically see in those 96 antibodies that we deliver to our partner. Since our libraries are mostly germline, with most of the diversity in the CDR3 of the light chain and heavy chain, we're really trying to get to broad epitopic diversity in this primary discovery delivery. And so we're really trying to find that functional epitope on the target in this initial delivery. And as you'll see in the subsequent slide, in that next stage of working with a partner, we can really home in by doing additional rounds of optimization to drive towards higher affinity. And so that's exactly what we do in that next step. After we deliver that set of 96 antibodies to the partner, they can assess them for function, really assess and see which set of antibodies has that right functional epitope. And then a nice real advantage of working with Adimab is that we can optimize a large set of antibodies in parallel. We can actually take in 6 to 8 antibodies of that original 96 and create libraries around them and optimize them in parallel, and then deliver back a panel of antibodies for each of those 6 to 8 leads. And as I'll show in the next couple slides, there's multiple strategies that we can utilize to then optimize those specific leads. And we can optimize them not only for affinity, but we can also do additional rounds of specificity tuning, or if need be, potentially optimize them for additional developability concerns. So how would we do that? Maybe in cycle 1, we might focus in on the heavy chain, CDR1 and 2 for an IgG lead, for example. And then maybe for cycle 2, if needed, if we need to really drive towards even higher affinity, we might focus in on the light chain and focus in on CDR1 and 2 and CDR3 of the light chain. And we would use that same facts-based workflow that I showed before to really drive either the specificity or the affinity as I showed before. Shown on this slide are just some potential affinity increases that we might see as we do that additional optimization. So shown in the open circles of one color is the original lead, and then the filled circles of the same color are the variants derived from that lead. And so as you can see, we typically can see affinity increases on the order of maybe 1, maybe even 2 orders of magnitude as we do the optimizations for both the IgGs as well as HCABs. Okay, so as I promised, that first room took a little while. Now I'll drive into that next room, and as I promised, now we'll talk about the HCAB library. So unlike the IgG libraries, which we have a lot of different heavy chain germlines as well as light chain germlines, the HCAB library, we primarily use the VH3-23 heavy chain germline framework as the backbone for our HCAB library. But we do incorporate multiple tetrads into framework 2 of the library. And then as I showed before in that previous slide, we do have a slightly longer heavy chain CDR3 diversity in heavy chain CDR3 as shown here on the right. We do incorporate some human heavy chain CDR3 diversity as well as some camelid-based diversity in heavy chain CDR3. Similar to the IgG-based libraries, we synthesize on order of the 10 to the 12th and create multiple copies of the libraries so that we never have to select the same library twice for the same target for different partners. And again, we also incorporate both immunogenicity and developability considerations into the design of the library. Okay, so let's move on now into the third room in the Adimab factory, which is humanization and optimization for our partners, which is actually a big component of our business as well. So a lot of partners come to us for bringing us a lead derived from some other technology, maybe derived from a mouse immunization they might do with another company, and they want us to use our yeast-based platform for doing humanization and optimization, which again, our platform is ideal for. So we have our own in-house software for doing humanization if the lead is derived from another species. So we can easily input that sequence into our in-house software, and then we can input the number of allowed amino acids back to mouse or back to some other species in both the heavy chain and light chain framework. And then it gives us a number of variants to then make and test, and then we can assess which humanized lead to move forward into optimization. And then once we have those humanized leads, and typically we'll pick about 2 or 3, as I'll show in the next slide, and then we can then take those leads and then move them forward to build libraries around them in our proprietary yeast to then select out high affinity variants from them. And so we have multiple strategies to build libraries in our yeast. Shown here is just one way that we'll typically do that. We'll then take our input antibody, and maybe we'll build a heavy chain library in one instance, maybe build a light chain library as a second library, and then maybe a third library we might build is a library we might fix some liabilities in the input antibody that we might get. And so in that initial cycle of selections, we might select them against the target, and then move forward all of the variants that we select for in that cycle 1 and combine them together in cycle 2. And then move forward all of the combined variants from cycle 2 together, and then merge them together with all of the liability fixes from the liability fix selection. Shown here is the results from that case study I just showed for one particular input antibody. It's kind of a busy slide, but on the y-axis are the affinities of both the parent as well as the variants, and showing the y-axis are the Hick retention times of the parent and the variants. What you want to look at are the symbols of the input parents, which are shown on the top here in the outline. So those are the input parents. And then you want to look at the variants, which have the same symbols, which don't have the outlines. And so what you can see is that all the variants go down on the graph, so they all increase in affinity. And then what you also can see is that all the variants move to the left in Hick retention time, which means that they're basically improving in overall developability, which as a course of both our discovery programs as well as our optimization programs, Adimab does a lot of developability testing in the course of our data packages that we give back to our customers. So you can really see that through the course of our optimization, we're not only improving affinity, but we're also improving the overall developability of these optimized candidates. Okay, now moving on to the fourth room of the factory. One thing I already did know a lot about Adimab before joining the company was our history and our expertise in developability through tons and tons of publications that Adimab had actually put out before joining the company, which of course I had read because I was a big fan of Adimab before joining the company. But what I did learn through the course of the year that I've been at the company is that we incorporate a lot of developability prediction as part of our projects that we do. So we have a lot of in silico tools for predicting developability based on primary amino acid sequence, which is a huge advantage of working with Adimab. So we can actually take in sequences that come out of our library, and we can predict, for example, Hick retention time, like those Hick retention times that I just showed on the previous slide. We can actually predict Hick retention time directly from primary amino acid sequence because we actually have models for Hick retention time from all of the discovered sequences that we've generated over the years. So because we have all this data, we can actually develop models for them as part of our overall workflow. And so I've been really blown away by how we've been able to incorporate a lot of these developability models, and we continue to be a leader in developability over the years, and we continue to publish in this area. Okay, so let's move on to the fifth room of the factory, which is our ability to do in vivo antibody discovery. So I spent a lot of time talking about how we have these amazing synthetic libraries, but what I learned is that we also can do in vivo antibody discovery. So did not know that. So basically what we can do is we can immunize both mice as well as llamas, and then we can take the output from those animals and then port them into our proprietary yeast and use our yeast as a way to do single B-cell cloning. So again, I didn't know we could do that. We could basically take advantage of the immune system of the animal, which is amazing, as well as take advantage of our yeast, which is an amazing way to basically screen out those populations of antibodies and basically get the best of both worlds. And so we have access, of course, to wild-type mice as well as llamas, but we also have access to the Ablexis system as well for humanized antibodies. So, and we typically try to reserve this workflow for really challenging targets like GPCRs and ion channels, where we really don't have access to recombinant protein, where it's more easily done with our synthetic libraries. So where we immunize these animals with either RNA or DNA-based sources of these antigens, generate immune response, and then we we can then port that into our yeast and then use either cells or other recombinant sources of these antigens to screen out those populations. And we can either use kind of single cell-based workflows or the FACS-based workflow I showed before to sort out those populations of antibodies derived from those immunizations using our yeast. Okay, and so shown here is just a little bit of data where we were able to do that for a really challenging GPCR target, CCR5. Shown on the left, is showing some data where we're able to find some really high affinity antibodies to CCR5 on cells. And then on the right, you can see that many of those antibodies were actually agonists of the receptor as well. As we heard in the previous presentation, finding agonists of GPCR is actually really challenging. And so in this case, we're actually able to find many agonists of this really challenging GPCR target. Okay, so let's move on towards protein engineering, and more specifically TCR engineering. One thing I've learned at Adimab is that if we can present it, in this case a protein, on the surface of our yeast, we can engineer it. And so, and as many of you probably know in this room, doing TCR engineering is not easy. But what we've been able to do at Adimab is kind of trick the TCR to present on the surface of our yeast. And the way we've been able to do that is we've been able to fuse it to the FC, much like an antibody, constrain it with a disulfide, as shown here on the left. And then sometimes we have to put a couple mutations in the TCR to enable it to express a little bit better. And then once we do that, and get good expression on the surface of our yeast, we can then build a library around it. So shown here on the right and in the middle, we've actually done this with this particular TCR against HLA-A2-NY-ESO1. Shown the red dot is the affinity for this TCR to this HLA, when as you can see, it's on the order of like negligible affinity, even micromolar affinity. And through multiple rounds of affinity maturation, multiple cycles of affinity maturation, we've been able to get this TCR to bind with picomolar affinity. We also did a deep mutational scanning library around this particular TCR as well, and we're also able to get it down into that picomolar range. And then finally, in terms of the number of mutations that was required to get it down to that range, it required about 9 to 10 different mutations. In comparison, another company, I won't mention their name, also did the same optimization, and they required several more mutations than we required. So in comparison, we actually required less mutations overall to get it down to that same affinity range. Okay, so in general, moving on to the next room, we also can just do general protein engineering. In this case, we had a partner that had a receptor that bound 2 different proteins, and they actually wanted to engineer binding away from one of those specific proteins. Again, as long as we can get expression of this receptor on the surface of our yeast, we can quite easily do that, and that's exactly what we did. We engineered this protein, this receptor, to express in our yeast, and then once we did that, we built a library around it, and then basically selected a way binding to that other accessory protein and kept binding to the one protein that we wanted to retain binding to as part of the overall engineering effort. Okay, so I spent a lot of time talking about both discovery of IgGs, optimization of IgGs, both from our synthetic libraries as well as our in vivo efforts. I've talked a lot about protein engineering, and more specifically even TCR-based engineering that we can do all in our proprietary yeast, which is, again, fabulous. Now I'm going to talk a little bit about some tools that we can utilize to basically complement all of those fantastic technologies. And so the first one I'll talk about in this next room are our heterodimerization technologies that allows us to create higher order bispecifics and multispecifics that might have multiple binding domains as part of the overall biologic that you might want to create. And so these are, again, mutations in either the heavy chain interface or the heavy chain light chain interface that you can then either license yourself, or you can get Adimab to use to basically create these higher order constructs for you. We also have access to our own, what we call our chain exchange mutations, that allow you to take 2 monospecific antibodies and bring together and create either bispecifics or multispecifics. and a really fast screening reaction. And then finally, because we have access to all these different bispecific and multispecific heterodimerization solutions, as well as our rich experience in this field, it really gives us a lot of diversity and flexibility to create all kinds of different constructs for you. Of course, we're always open to making new constructs that you might want to create. So again, you can always come to us with any new ideas that you might have. Okay, so let's dive into it a little bit more. So in terms of our heavy chain, light chain pairing solutions, we have mutation sets between CH1 and constant kappa, where the mutations are on the CH1 side. We have mutation sets between CH1 and constant lambda, where again, the mutations are on the CH1 side of that interface. And then we have mutations between CH1 and C kappa, where the mutations are on both sides of the interface. The advantage of all of these mutations is they're all in the constant domains of the Fab region. They're not in the FV region, so they're very plug and play. They have no impact on the overall combining region of the Fab. And if you want any more information on these mutations, we actually published a really fantastic paper earlier this year in MABS that I would refer you to. In terms of our heavy chain pairing solutions, I did talk a little bit about our chain exchange mutations, which allow you to basically take two monospecific antibodies that have these chain exchange mutations built into them and basically use them to basically rapidly screen a large number of bispecifics and multispecific combinations. We also have our own version of kind of knob and hole based CH3 based mutations that allow you to create a kind of scale up version of your final bispecific or multispecific. And again, these two sets of mutations work really well in tandem with each other, one for initial screening and the second for kind of your final scale up of your bispecific or multispecific. Okay, so the last technology room I want to highlight are our CD3 binders, which again work really nice in tandem with the heterodimerization solutions I just talked about if you want to create your own T-cell engager. And so the first one is our CD3 IgG binder that we generated that has human cyno cross-reactivity to CD3. We also took this same CD3 binder and optimized it for a wide range of different affinities as shown here on the bottom left. And so as you can see, we've tuned it for a wide range of different affinities from almost picomolar affinity all the way to micromolar affinity. And so this is really nice because you can basically tune your T-cell engager to your specific tumor antigen binder. And so as you're building your T-cell engager, you can kind of pick out the exact affinity of CD3 binder that's exactly purpose-built for your T-cell engager as you're constructing it. Shown here on the right is just a little bit of data where we took two of those CD3 binders and paired it with one specific CD20 binder. And as you can see, we're seeing really potent killing that's comparable to the Genentech CD3 binder, mosunetuzumab, in a 1+1 bispecific T-cell engager. And then finally, we also have our own CD3 binder that's in an HCAB format that was derived from a llama immunization. Unlike the CD3 IgG binders that I just showed that had human cyno cross-reactivity, these HCAB binders are very human-specific. We're still trying to find some VHH or HCAB binders that have human cyno cross-reactivity, but for the time being, our HCAB binders for the time being are very human-specific. But they do have really nice binding to human CD3 with a wide range of different affinities for the panel that we have. They have great developability as shown in the middle panel. And then in 1+1 bispecifics as shown on the right, those wide range of different affinities gives you a wide range of different killing when paired with different tumor antigen binders. Okay, so I'm gonna go back one. And then the last room, I'm just gonna end with the people at Adimab, the people that basically created all these amazing technologies I just talked about. And again, I've just been blown away by all the people, all my colleagues that I've been working with this last year. One thing that I've really learned to appreciate is that the people that I work with have like a lot of experience that they bring to the table. in terms of antibody engineering, protein biochemistry, computational biology, high throughput expression and analytics, and computational biology. There's I'm just showing you some statistics here. There's people that have been at the company, some people have been there upwards of over 10 years. So, and a lot of these people come to the table and work on the projects with you on a day-to-day basis. So not only do you get access to all these amazing technologies, but you also get access to these amazing people that come along with it. So really been blown away by working with these people day in and day out, but as well as if you get to work with Adimab, you also get to work with all these amazing people as well. Okay, so just to conclude, Adimab is a premier therapeutic antibody company. We're really focused on not only creating all these fantastic technologies that you get access to, but also really focused on incorporating developability into every facet of what we do. We have all these amazing discovery libraries, both synthetic as well as in vivo-based libraries. We also can do antibody optimization as well. And then we can also do protein engineering, both TCR-based as well as protein engineering as well. And then we have a whole toolbox of other technologies to allow you to create higher order bispecifics and multispecifics, as well as T-cell engagers with our CD3 panel, both IgG as well as HCAB-based. And then finally, last but not least, the people that have come up with all these amazing technologies, as well as our ability to employ them to help you discover the next generation of biologics for the future. So I'd like just to end with thanking you for your time and attention today. Shown here is my email. I know I kind of went through everything at a really high level, but I have really detailed information about everything I talked about today. So if you want any more detailed information about anything I talked about today, happy to give it to you. So I'd like to thank you for your time and attention today, and I'd be happy to answer any questions.